Okay, so our objective today is to describe different characteristics of viruses and compare the structure to that of the cells we already know. We will depict our own viruses in models. Obviously, if you're watching this, we've already done it, or you're excused because you were absent. So what we're going to talk about over the course of these virus lectures is what are viruses? Are viruses living things? Are there different types of viruses? How do viruses reproduce? And how do viruses cause disease? So this is a virus. I'm not sure what it's to, but it is a virus. You can see all of these, the blue shiny things right here. These are glycoproteins. And if you'll remember, our cells have membranes, and in those membranes we have receptors. These glycoproteins attach to those receptors, so these help the viruses bind to our cells. So this is also a spherical virus, and it's going to have the genetic material inside of it, the envelope, and the glycoproteins on the outside. That's it. So these are viruses, more viruses, just, another kind. Pretty color. This is also a virus. This one looks very scary and deadly. This is another one. This one kind of has a sticky look to it. A couple of classes said it looked all snot covered and gross, so I agree with that. Later in the slides you'll see a picture of the red blood cells, which are these, which have been lysed open and viruses like pouring out of them and stuff. It's pretty interesting looking. So this is a bacteriophage. It's specifically a virus which attacks bacteria. So these are not legs. They're called tail proteins, and they just help the virus cell attach to the cell, or to the bacteria cell. All of this outside part is called a capsid, and that's just the protein coat that surrounds the virus. The virus's DNA is going to be up in here. This is called the head. And so this is an electron scanning micrograph, and we'll talk more about these a little later. That's just what the picture was taken with. So what are viruses? They are microscopic particles capable of infecting living cells and inserting the genetic material inside. So this is the book's definition, and this is a more relaxed definition I found. So genes in a box. It's infectious particles consisting of a nucleic acid wrapped in a protein coat called a capsid. So that's all it is. So are viruses considered to be living things? Are they alive? No, they are not. They're not cellular, so they lack organelles. So what does this mean? So they cannot grow, they cannot perform metabolism, no cellular respiration, nothing like that. They also cannot reproduce on their own. Very key thing in determining whether something's living or not. So it has to have the host cell. The host cell provides most of the necessary replicating, transcribing, and translating components necessary to copy its DNA. So this part down here, only worry about this if it helps you understand. If not, don't worry about it. So it's just talking about how they're passive little things, completely at the mercy of their environment, controlled by their genetic material. They do not do anything. Nothing out of ordinary, nothing like that. So again, here's a bacteriophage. This is just a model. So the little tail proteins, the DNA is up in here. And what it's going to do, it's going to hook onto a cell. And then basically it's going to poop its DNA into the bacteria cell. Alright, that's what's going to happen. Alright, so this is a spherical protein. Or not spherical protein, I apologize. Spherical virus. So you can see it has a nuclear or an envelope around here. And then this would be the capsid. Some spherical proteins have a capsid, some do not. And then the genetic material is in here. Here are the glycoproteins. So these are the different parts of a spherical virus. So are there different types of viruses? Viruses differ based on their structural makeup. You can have spherical, which we just talked about, so membranous outer envelope with projecting glycoprotein spikes. You can have a helical virus, which is capsid and the proteins wind in a helix like this. You can have polyhedral. So polyhedral is the same shape over and over again. So like a cube is a polyhedral. Viruses, it's going to be more diamond shapes or triangle shapes and stuff like that. There's some examples on the next slide. So a combination, this is going to be the bacteriophage. So the bacteriophage, it has a polyhedral head with a helical body part. So here's our spherical. And then this is going to be our polyhedral. So each of these circles represents a protein, and these protein subunits are going to form this same shape over and over again. So do you see how this is making triangles? And then the DNA is all inside of here. So here's helical. All of the proteins are in a helix. And then the genetic material, the DNA, is going to be inside of there. So here's the bacteriophage. So this is the head. And do you see how it's polyhedral, the triangles? And then this is helical. So we're going to call that combination. Don't worry about this word, binyl. That's silly. So viruses also differ by the type of genetic material they possess. So if it has RNA, it's called a retrovirus. This is a very important word. Remember it. So viral enzyme uses viral RNA as a template to make cDNA, or complementary DNA, from which more virus is made. So basically what this is saying is you'll have a virus. This will just be my spherical virus, and it's going to have RNA in it. It also has a little enzyme called reverse transcriptase, and this takes RNA and reverse transcribes it to DNA. So then you end up with your DNA strand, and this is what is replicated by the host cell. So when you go backwards from RNA to DNA, the DNA is called complementary DNA. That's just, the proper name for it. So the genetic material, they may also have DNA. So I didn't put anything here because we know what DNA is. We're pretty familiar with that. That's just the commonplace genetic material. Alright, so this is the last slide, and don't worry too much about this. We're going to go over it more in the next lecture. This is just an overview of cellular or viral reproduction, so how a virus is going to reproduce and infect a whole bunch of cells. So we have the lysogenic cycle and the lytic cycle. The lysogenic is a dormant cycle. This is where it's just going to replicate and replicate and replicate. You're not going to show any symptoms or anything like that. The lytic cycle, where it's lysing, this is actually going to cause cell damage, and that's where you're going to see tissue damage and stuff like that. This is where you're going to see symptoms of the disease you have. So let's start up here. So here's our little phage. She's going to poop the little DNA in here, and you're going to end up with a viral DNA and a bacterial chromosome. So these two combine right here, and each time this bacteria cell replicates, it's going to replicate the viral DNA as well. So you're going to end up with hundreds of cells that are each infected with this viral DNA. So once again, there are no symptoms right here. It's just replicating, just replicating. It's kind of hiding back in your body. So here's one of our replicated cells. It has the viral DNA inside of it. So if you've heard of herpes before, I am sure you have. Herpes is a sexually transmitted disease, not all the time, but usually. And once you have herpes, you always have herpes. So when you aren't showing signs, that means the virus is in this lysogenic cycle. It's just going to be replicating, just going to be replicating. When you have cold sores or genital sores or anything like that, you know that the virus is in the lytic cycle. So how does it go from the lysogenic to the lytic? This viral DNA right here is going to be activated. In the case of herpes, it's by stress. And so when you get really stressed out, this viral DNA is going to come back out of the bacterial DNA. and it's going to make a whole bunch of new viruses. So it's just going to replicate over and over and over into actual viruses. These viruses are going to end up breaking out of the cell. This is called lysing the cell. And they're each going to go and infect new cells, and this whole thing starts over again. Okay? So that's it for this lecture. And I'll go ahead and put the next one up too. Oh, actually, that's not it. Sorry, if you're still listening. Here is that picture I was talking about. So here are the red blood cells. And then these are red blood cells that a virus has attacked. And so this is the virus. And you can see that it's all spilled out. And this one looks like it's about ready to lyse. And all of these green viruses will come pouring out to go infect other cells. Anyway. Okay. So here's the model. Again, don't worry about this. This is old news. We've already taken care of this. Okay, so today we talked about the lytic and lysogenic cycles, so viral reproduction, and then we worked on understanding how viruses, prions, and viroids cause disease. So another topic we talked about was how viruses come to be. So how we have one virus and it mutates into another virus, or it forms a whole new virus altogether. And these, oops, that's not what I wanted to do. So these links right here, this top one is the movie trailer for Contagion, and this is the last minute and a half of that movie. It's a pretty cool movie, as you know, it's all about disease transmission and stuff like that. So if you have time to watch it, or the whole movie, it's pretty cool. If not, just try and watch these links. This is going to show you that this virus came from a bat and a pig. And the two viruses from them mixing, and so it mutated into this cross-species virus, which then affected humans and almost wiped everyone out. So, anyway. Alright, so here are the lytic and lysogenic cycles that we talked about in the last lecture. So, I'll go over them again. So, we're going to start up here. So, here's our phage, and he's going to poop his DNA in, and then also, the viral DNA is always going to form a circle. It's much easier to copy a circle than it is a line, because the circle, it can just go around and around and around and around, whereas if you have a strand of DNA, it has to get back to the front each time, and it has to most likely use new enzymes each time. So, anyway, at this point, it's going to determine whether it's going to go into the lysogenic or the lytic cycle. If it goes into the lysogenic, this viral DNA is going to join this bacterial chromosome. So once it does that, we call the viral DNA a prophage. So you'll need to write this down. A prophage is viral DNA once it has joined with the bacterial chromosome. So once this has happened, every time our bacteria cell replicates, it's going to replicate our prophage as well. So you're going to end up with hundreds of different cells infected with the viral DNA. So once again, the lysogenic cycle is the dormant cycle. You're not going to see symptoms here. This is just, it's replicating, it's staying in the background. So like in herpes and chicken pox, once you have those viruses, you will always have them. It's just whether or not it's in the lysogenic cycle or the lytic cycle. So here is our cell that's been infected with the virus. So there's its viral DNA. And once this viral DNA is activated, usually by environmental stress, it's going to pop out of the bacterial chromosome, and it's going to start replicating itself into new viruses. So once again, once all these viruses fill up the cell, it's going to burst it. It's going to lyse open the cell, and all these new viruses are going to go out and infect other cells, and the whole process starts over again. So you can kind of see how a bacterial cell could come in, and it's going to follow the lysogenic over and over and over again, and then it can lead right into the lytic cycle. So lytic is where you're going to see actual symptoms. This lysis, the cell lysis, that's going to be actual tissue damage. So in herpes, when the cells start lysing, that's when you're going to see the fever blisters and the genital sores and all those things. Alright, so here again is the lytic cycle. So bacteriophage, DNA inside, empty phage shell leaves, and then this bacteriophage DNA, the viral DNA, is just going to replicate over and over and over again, and it's going to code for these pieces of the viruses, which are going to be rejoined. It's going to be assembled into new viruses, and then these viruses are going to lyse out of the cell and go and attack new cells, and then the process starts over again. So here are the actual written out definitions of what's going on. So the lysogenic cycle, this just fits so well into this picture, I didn't want to separate the two. So the lysogenic cell, remember, that's just the dormant. It's going to join with the bacterial chromosome and become prophage, and then it's just going to replicate with the bacteria cell every time. And then the prophage is going to become activated by stress, and it's going to pull out of the bacterial chromosome, and that's when it starts the lytic cycle. So here are the actual steps of the lysogenic cycle. Okay, so here's where we start talking about the two clips you watched at the beginning. How do new viruses appear? So viruses that appear suddenly are called emerging viruses. This can be caused by contact between species, so like the bat and the pig from the clip. You can have, it can spread between isolated populations. So this was the HIV virus. It was pretty contained in different places in the world, and then we had, travel and everything where everyone was all up in each other's business, and HIV spread to now it's a worldwide epidemic. So then you can also have mutations. This is why we have to get a new flu shot every year, because the flu virus mutates so often. There are so many different strains of the flu virus. So from mutation, do you think a DNA virus or an RNA virus, a retrovirus, would mutate faster? Okay, so it's going to be an RNA virus. And this is because there is no proofreading or correcting system like in DNA. So remember when we talked about cell cycles, when the cell was replicating its DNA, it had an enzyme called exonuclease, which followed behind it, and it made sure that there were no incorrect nucleotides put into the DNA strand. When we go from DNA to RNA, there's the proofreading. When we go from RNA to DNA, there is no proofreading system. So if an RNA virus picks up a mutation, it's stuck with it. So these existing viruses can evolve into new strains, which can infect those previously immune. This is why we need the new flu shot. So every year, there are multiple strains of the flu. Doctors high up somewhere, I don't know where they are or who they are, they determine which virus they think is going to be most prevalent or most active, and that is the flu strain that we have a vaccine for. So that's why you can get the flu and then get sick with another version of the flu, because you're not vaccinated against all the strains at once, just one at a time. So you do not need to know this AIDS stuff. I thought you did, and you don't. So this is just kind of listen. It's an example of what we've been talking about. So what's the difference between AIDS and HIV? AIDS is going to be the disease. HIV is going to be the virus. So acquired immunodeficiency syndrome, and then human immunodeficiency virus. And the thing that makes HIV special is it has 2 copies of its genetic material instead of 1 copy. So AIDS is going to be a spherical virus. So earlier we talked about how sometimes spherical viruses can have a capsid. They don't always have to. HIV has one. So it does have a capsid. And here are the 2 copies of its RNA. So it's a retrovirus. And here are the two enzymes, reverse transcriptase, that are going to take the RNA and transcribe it into DNA. So here's where it actually shows what's going on. So here's our RNA, here's our reverse transcriptase, and it's going to copy that into DNA, and it's going to make it double-stranded. So then this helix is going to be it's going to join with the host chromosome, and then here is where the host chromosome takes over. So the host cell is going to use its own mechanisms and its own enzymes to copy the HIV virus DNA back into RNA. And then this RNA is going to code for all the different components of the HIV virus, which it's then going to complete and put together and package off and send to other cells. So like I said, you don't need to know the details of this. It's just for your information. So here's the steps. If you want to copy those down, you can. Alright, so you do need to write this. Viroids and prions. So viroids are small circular RNA molecules infecting plants. They are just RNA. There's no capsid, there's no envelope, it's just a circle of RNA. That's it. So they cause errors in the regulatory systems controlling plant growth. And this is going to cause abnormal development or stunted growth. Prions, you might have heard before, it has a lot to do with the mad cow disease. A couple of years ago, England, they were packaging off and sending meat that was infected with mad cow disease, and it caused a really big problem. They had to cull, I don't know how many cows, but anyway, it was sad. So a prion is a misfolded protein, and these misfolded proteins usually clump together. So in the proteins that, misfold are brain proteins. They are found in the brain. And this causes degenerative brain diseases in animals. You do not need to know these different examples. They're just examples. Scrappy is in goats and sheep. Chronic wasting is in deer and elk. Mad cow disease is in cows. And then Creutzfeldt-Jacobs disease is the mad cow version for humans. So. And I think that's it for that lecture, and I'll go ahead and try and put the next one up as well. So this is going to be the lecture for Tuesday. We're going to start our lectures on prokaryotes. So prokaryotes, this is going to be bacteria and protists. Right now we're just going to talk about bacteria. So we're going to do a disinfectant lab today, finish that up on Thursday, and then take some prokaryote notes. So these are different pictures of plates that have been streaked with bacteria. It's growing different bacteria colonies. Some of them are also growing yeast or mold, all of these different things. So again, more slides, more pictures. So this is most likely a mold. It kind of looks a little bit like penicillin. These little sticky looking things, those are bacteria colonies. This is an agar plate, but it's sheep's blood agar. So this bacteria feeds on blood, and that's what's in this agar. That's why it's red. So that's pretty cool. Alright, so prokaryotes are very, very tiny. I don't know why it's cutting off part of the slide, but anyway, just pretend it's not, I guess. So this is the tip of a pin, like a little pointy pin that you stab people with. And then this is an individual bacterium, this is an individual bacterium, this is an individual bacterium. So these are clumps of bacteria. So that's how tiny these little prokaryotes are. So, cell shape. So bacteria, it's either coxy or spherical, bacilli, which is rod-shaped, or spirochete, which is spiral. So, spiral, rod, and then coxy. And the coxy, if it's strepto, this prefix, that means that it is in strips. They are connected like this. And then, so this is what causes strep throat, the bacteria. And then staph is going to be a clump of circles, this clump of little circular bacteria, like this. That's going to cause staph infections. You can also have single little bacteriums. So then bacilli, which are the rod-shaped, they're usually singular. This is like E. coli. And then spirochete, which is spiral-shaped, that is what the syphilis virus or syphilis bacteria is. So cell wall, you can have gram-positive, which is this gram-positive bacteria. It's simple cell wall with a relatively thick layer of peptidoglycan. This is just a way to differentiate between bacteria. And then gram-negative, it's going to be a more complex wall with less peptidoglycan. It's very important to know this. There might be a question on the test. Hint, hint. So gram-negative bacteria are usually more serious. They're more threatening. They're often toxic. So over here, you can see that this bacteria is gram-negative because it's pink. I don't know if I put that on there. Gram-negative is pink. Gram-positive is purple stain. So here is gram-positive. Okay, so cellular adaptions, external structures. So you have a capsule. Some of them have a capsule. It's a sticky layer allowing them to adhere to a surface or others in a colony. Flagella, structure allowing them to move around in response to environmental signals. So here's a bacteria cell, and then this flagella. It's just a protein structure that can flip around, and it kind of propels them forward. So environmental signals, this would be they can move towards food or away from toxins. Fimbriae, same thing as pili. These are hair-like projections enabling some to attach to surfaces. So like gonorrhea, it has fimbriae. That's how it attaches to your reproductive tract. Alright, so here is the capsule. These are just pictures of what we talked about. So sticky coating, and this is actually, I don't know what's happening to the top of these slides, but this is this epithelium of your tonsils, so the skin cells of your tonsils, and this is a bacterium that's sticking to it. And then here's our bacteria. So here's our fimbriae, or cilia, or not cilia, sorry. These are fimbriae or pili. And then here's the flagella. Alright, so some features and adaptations of prokaryotes. Most replicate within 1 to 3 hours. Their genome is one circular chromosome. So this one circular chromosome is huge. So it's just wound up super tight and like a knot inside the bacterium. I'll show you a picture in just a minute. So each replication can give rise to spontaneous mutations, which leads to lots of variation. There are so many different types of bacteria, different types of prokaryotes. So, anyway. So plasmids, these are outside this genome. They're small circular DNA molecules which replicate independently. So they can carry genes to enhance survival, kind of off on their own. So resistance to antibiotics, or the genes to produce an endospore, which are resilient cells. It kind of, here's our bacteria cell. And so this circular little plasmid is going to start up an endospore, which is going to be really dark. And this outer cell is going to disappear. So it goes away. And then you're left with this endospore, which has the genetic material in it, and it can withstand really hot temperatures, really low temperatures, and it sits there and it waits for the environmental conditions to stabilize, and then it can rehydrate and turn into a bacterial cell again. It's pretty cool. Alright, so here is the picture of the bacteria. So this all is the same, all of this is, it's one circular chromosome. It's all connected. These little guys are plasmids. Do you see how they're completely separate? They're little circles off by themselves. So you have plasmids and a chromosome. One giant chromosome. So here's a bacteria cell. It's been ruptured. That's why its DNA is spewing everywhere. So this is a bacteria cell with an endospore in it. So these layers, this part of the cell is eventually going to disintegrate and disappear, and you're left with just the endospore. And I think that's where we're going to stop tomorrow. If I get a little further, I'll put the rest of the notes up tomorrow evening.